Thank you so much, John. It's a real pleasure to be here. Thanks for having me. I've had a great few days at the Biostatistics unit and a very good time today listening to some really interesting talks, which I feel like several of which have kind of acted as helpful motivations, if you like, for some of the things I'm going to say here. We've heard a few things about latent factor models of various kinds, bi-clustering, and also a bit about, I think, even in the causal inference talk on, I don't know, to trying to understand what's going on as being a kind of a high-level goal. And that's how I see matrix factorization methods, if you like, as a tool in the statistical toolbox to try to help us understand what's going on. They're really, I think, exploratory data analysis tools, analogous to, say, clustering being a data analysis tool. So the last talk talked about the connection between clustering and bi-clustering and matrix factorizations, which we're going to talk about more here. So with that stage set, let me just tell you what I mean by a matrix factorization method. So given a data matrix X, which is measured on, say, n samples and p variables, and so I know in some of my examples later, n will be the cells and p will be genes, and xij will be a measure of the expression of gene j in cell I. But you could think of your own examples where you might have a data matrix, and matrix factorization methods attempt to find usually smaller matrices L and F that have k columns. So k is going to be essentially the number of different, or the affect the complexity or the rank of this factorization. So we're going to look for L and F such that X is approximately equal to LF transpose, and there could be many different ways of formalizing what we mean by approximately equal to here. But if you just want, just for an example, we could try to minimize the sum of squared errors here. So that's just an example of how you might go about looking for L and F. And importantly, we might consider different types of constraint on L and F. And two widely used examples of matrix factorization methods are principal components analysis and non-negative matrix factorization. And these two different methods correspond essentially to different assumptions on L and F. So if you assume that L and F are orthonormal or orthogonal, then you essentially get back principal components analysis. And if you assume that L and F are non-negative, that is, every element of L is non-negative, every element of F is non-negative, then you get back this method known as non-negative matrix factorization. So there's a whole menagerie, if you like, of matrix factorization methods out there that correspond to different assumptions on L and F. Okay, so a kind of trivial but important observation is that you can write this factorization, L times F transpose, as a sum. Okay, so this is just a different way of writing a matrix product as a sum over the columns of LK, FK transpose, where LK is the kth column of L and FK is the kth column of F. So this is just writing, it just says that if X is LF transpose, then you can write X as a sum of K rank 1 matrices. Another way of thinking about this, I guess, is that the Fs are forming a basis for the rows of X. So in that interpretation, the Ls are the weights on that basis, and you write the ith row of X as a sum of linear combination of the basis vectors F. And you can also think of L as a basis for the columns of X. So, okay, but for now, let's just go back to this decomposition. So sum of k things, and sometimes these k things are somehow interpretable, either scientifically or physically. And so sometimes this idea that you've decomposed, if they are interpretable, then you've decomposed X into a sum of k interpretable parts. And Lee and Sung used the phrase parts-based representation to kind of refer to this idea that you're decomposing X into I mean, the interpretable is kind of maybe implicit in the parts. So I guess, to set the stage a little bit, this is this is a different way of thinking about matrix factorization than purely an embedding idea, right? So if you've come across the idea that PCA is creating an embedding, it is. And but in order for it to be considered a parts-based embedding or decomposition, the bits have to be individually interpretable. So in terms of if you've got a k-dimensional embedding consisting of k numbers, each number has to mean something essentially for this which isn't generally required of an embedding, right? And in fact, principal components analysis is a great way to find embeddings. It's very simple, quite effective, but it's not a great way to find parts-based representations. That is, when you ask questions like, what's PC1, what's PC2, what's PC3, etc. If you're lucky, PC1 might correspond to something you can say. If you're really lucky, PC2 corresponds to something you can say, and then pretty much you're not going to have any luck after that. So, okay, so that's going to be a kind of a recurring theme, is trying to understand what these parts are and what assumptions lead us to parts that, or decompositions that are interpretable, essentially. So having thought about this a while, worked on it a while, these are 4 things. Maybe it's not exhaustive, but these are 4 things I think can impact the decomposition you get and how interpretable it is, or how it is interpreted. Okay, so non-negativity, actually, Lian Sung's original paper on NMF was really trying to make the case, I think, that assuming non-negativity tends to lead to more interpretable parts-based decompositions. We'll see their example in a minute. Assuming sparsity on LNF can help interpretability. How you transform the data, or whether you use some kind of link function, as I'll show later, affects the results for sure, and arguably perhaps the interpretability of the results. And whether or not you assume orthogonality on the L's and F's can also have an impact. And yeah, maybe on just one rather than both. Okay, so this is Lian Sung's motivating example to try to get, it's a non-genetic example, but it's quite a visual example. Essentially, their data set was images of faces, grayscale images of faces. So each face is, of course, a pixel map with a number for each pixel. So what they're doing is they're vectorizing that. So you take the pixels in a face and you create a row of a matrix using those pixels. And then you take another face and create another row. So your data matrix X, the rows are images or faces, and the columns are pixels, okay? And then you seek well, in this case, a principal components analysis. You do a principal components analysis of that matrix. And what it ends up doing is these are the essentially the Fs that you get out of that decomposition, but rearranged. Each F is a row of that is a row, but we rearrange them back into the shape of a face in order to visualize them. Oh, sorry. So what you see is that the first PC is essentially an average face. Yeah. And then subsequent PCs, this one's a nice one. I like this one. It's a contrast, if you like, between eyes and the rest of the face somehow, right? So arguably somewhat interpretable, but generally kind of hard to interpret, right? We can pick out a few that we might be able to say, oh, it's a contrast between eyes and everything else. But generally, the PCs are not necessarily very easy to interpret apart from this one. Okay, so the idea is that you can represent each face in your database or in your data matrix as a linear combination of these, quotes, eigenfaces, and you get a weight on each eigenface, which are kind of visually represented here, which can be positive or negative. So importantly, in PCA, things can be positive or negative. So black is positive, red is negative here. And I think part of the argument of their paper is that actually makes things a little harder to interpret, or maybe a lot harder to interpret. So when you do NMF on these data, you instead get something like this. It's still decomposing the faces into a basis, but the bases are now non-negative. And so what ends up happening is that you kind of see parts of a face in each basis vector, or at least that's their argument, right? So these are the eyebrows I don't know, is this a mouth or a chin? I'm not quite sure. Is that a mustache? Maybe. Anyway, so that's their argument. That's what they mean by NMF tends to lead to parts-based representations. And, you know, one of the reasons maybe it's more interpretable, you can certainly see that the non-negative assumption leads to sparser basis vectors. That's because sometimes you, like when you're fitting these things, it wants to make something negative, but you're not letting it, so it sets it to 0. And so you get sparse, even without any additional sparse penalties, you tend to get somewhat sparse representations here. The other thing that's going on here is because everything's positive, you can never have things canceling out with one another. You're creating a face by adding up things that can't cancel out with another. They have to add. And that somehow, avoiding that cancellation somehow makes things easier to interpret. That's maybe an intuition. Okay, so to give a genetic example, what happens if we apply NMF to genetic data? Well, here's an example. Okay, I should maybe say NMF is also comes under there are various words that are used. In population genetics, people use admixture models. They're essentially a version of NMF. In document analysis, people use topic models. They're also a version of NMF. I'm going to call them all NMF for now. But one version of NMF uses a Poisson likelihood, and that's quite natural for count data. So what we're analyzing here is data from the GTEx project on gene expression, where this is not single cell data, this is bulk data. So each sample is a piece of a human tissue. And there are about 1,000 different donors with 50 different human tissues. So we're not worrying about who gave what. We're just stacking those in rows. And the columns are genes. And so Xij is a count of how many times was gene J seen in sample I. And the size factor here represents how many total counts did we get from sample I, because some samples produced more sequences than others. So you can think of that as kind of a technical factor we want to take care of. So I've added that here. And then the key part here are the LF transpose part. So just to, L and F are non-negative, and you can think of them in terms of interpretation. You can think of LIK as representing what's the membership of sample I in this kth component. And you can think of FJK as representing the relative expression of gene J due to that component. And okay, so what do the components represent? Well, who knows? Let's see what we get, shall we? So what I'm going to show you now is a picture of just the L's. I'm going to focus on the L's. So the memberships of sample I in each component. I'm going to color each component in a different color. Oh, before I do that, I'm going to show you a different representation of the data, actually, which is a two-dimensional t-SNE embedding of the data. If you've not seen t-SNE before, it just tries to put all the samples in a two-dimensional space such that samples that are clustered together in the high-dimensional space also cluster together in the low-dimensional space. And so what you can see here is the samples are colored by tissue. And so you can see there are some tissues, I don't know what each color is, I'm sorry, but you can see some tissues that are all very clustering together. And then you can see other things which are a bit more blobby, but maybe that's not surprising. Some different tissues are quite similar to one another in these data. Okay, but this is a two-dimensional embedding that helps visualize the data. So I guess I want to say, this is an alternative way of visualizing the structure in this data, that this is the result of the NMF. So each column in this picture is a sample, and then I've colored them according to their membership in the different components. And there are about 20 different colors, I think, in this picture. So 20 different components. We chose 20 by how many different colors can you manage to discern? These pictures do get kind of complicated if you try to use more than about 20 or 30 because colors overlap. Okay, so when I say a column here, I mean a tiny, tiny column that's very thin. And then the columns are arranged according to which tissue they came from, right? So all these tissues are from skeletal muscle, and there's lots and lots of columns here if you look closely. But they all have very strong membership in this kind of orangey cluster, right? Or component. Okay, I use the word cluster. Why do I use the word cluster? Actually, what's kind of emerging from this picture is already a kind of a bit of a cluster, right? Because most samples have one dominant color, and then not many others either, right? So, most of these samples from these tissues are lilac, but they also have a little membership of this dark purple cluster component. So the way I think of this representation is as kind of close to a clustering type representation. Maybe there are some samples here which are not quite, they're obviously being represented as some kind of mixture between two clusters, which might be a bit more obvious in this representation than in this one. What's, I think, particularly interesting is the potential here to see for the representation to highlight similarities between two groups. So this is the tibial arteries, and these are some smooth, well, these are actually from the esophagus, but when you look at the genes involved in this salmon component, you see it's genes that are involved in smooth muscle. So what you're seeing is that this component here that's common in the esophagus is also present in the artery here. And that makes some sense biologically because arteries actually use some of these smooth muscle genes to kind of help propel things through them. So that's a thing that is very difficult to see from a picture like this. So although it's at a high level, roughly giving you something like a clustering of the data, it's also got the capability to highlight similarities or something in common between two different groups that might be lost. If you just clustered the cells, you get clusters and you lose the information, that there's some kind of maybe somewhat subtle similarity going on between these samples and these samples that doesn't maybe correspond to the global clustering structure. On the other hand, maybe we'd like a bit more of this. There must be a bit more of this going on, right? So some of the representations I'll show later are maybe a little bit less clustered, a bit more complicated, but as a result have the potential maybe to highlight some of these similarities that are a bit more subtle more often. Okay, so that was non-negativity. I want to go on now to talk about sparsity. So sparsity is another thing that can help make these decompositions interpretable. And my example here comes from it's also the GTEx data, but the data matrix now is a different data matrix. It's a data matrix on eQTL effect sizes in different tissues. So the rows now, each row is a gene SNP pair or a SNP gene pair, and the columns are the tissues, and the ijth entry is what's the estimated effect size of that SNP on that gene's expression in that tissue, right? So they're EQ. So the idea here is that matrix tells us something about what the patterns of sharing of eQTL effects across tissues might look like. So that's what we're going to try and extract from the data. So Xij is the observed effect size of eQTLI tissue J. LIK is the quotes, and I've put it in quotes now because LIK can be negative. So membership is kind of more naturally used when things are non-negative, I think, but just the membership of eQTLI or weight of eQTLI for component K, and FJK is an effect size in tissue J due to component K. So this time, I'm actually going to show you some of the Fs. Right? You can visualize both things and go looking at both things, and both can be useful. Just going to focus on the Fs this size. So these are going to be vectors of length, the number of tissues, right? Each F is a vector of length, the number of tissues. And these are the ones we get out. Let's look at the first one first. The bar plot here has a size for each tissue. The tissues are arranged in alphabetical order, which means all the brain tissues are together in yellow here. And yeah, so is that true? I think I feel like there's some things missing on the bottom actually. Now I say this because these colors aren't there. Sorry, I think there must be some other things here. Anyway, so you don't need to worry about too much, but I think knowing that the yellow one's a brain is helpful. So focus on that for a moment. So this is the first factor. It's also pretty much the first principal components, actually. It's not sparse. You can see that our method didn't end up it doesn't force things to be sparse. It allows things to be sparse, encourages it when necessary. I'll say a bit more about the method shortly. But what this is capturing is the fact that there's a lot of sharing of EQTL effects across tissues, that to a first approximation, if an EQTL is an EQTL in one tissue, it's an EQTL with actually somewhat similar effect sizes in every tissue. The second factor or component captures essentially the fact that the brain tissues often or sometimes show a different effect size than the other tissues. And that's also a feature of the data that you can see fairly easily looking at other methods. And then these subsequent factors, which are a bit more sparse, some of them are capturing what you might call tissue-specific effects. So this one's actually whole blood. It's not listed here, but this is whole blood. So it turns out that the whole blood sometimes shows a very different effect size than all the other tissues, maybe blood-specific type effect size. So this is a factor that's capturing those kinds of things. Maybe more interesting, this is one that's capturing essentially these two tissues here are the sun not exposed and the sun exposed skin tissues. So there's two skin tissues in the data set. Some are sun exposed, some are not sun exposed, depending on where they were taken from the body. So maybe it's not surprising that they show some greater sharing of effects than other pairs of tissues. So a EQTL that's loaded heavily on this factor is showing a very different effect size in the two skin tissues than it is everywhere else. And there's another tissue here, I think is esophageal, that seems to share something in EQTLs with the skins. These are, I'll skip over those. Let me skip to some other one. This one's two heart tissues here. So when I see factors like this, that makes me think I'm picking out something real and interpretable. They're the two heart tissues. These are the two cerebellar tissues within the brain. So this is capturing the fact that sometimes the cerebellum shows some specific effects. Okay, so just to compare with a non-sparse version, which essentially is PCA, but it's actually, we use the software soft impute. It's like, it's a slightly regularized version of PCA, but without any sparsity. You can see the first factor is the same as ours, essentially. The second one's pretty similar, but not quite so sparse, but it's still capturing brain versus others, although there's a few things going with the brain here. And then everything else is, I don't know what's going on. I mean, it's an eigenfunction of some covariance matrix, right? Essentially, that doesn't really have this interpretation the same way. It's not such a part-space representation as the one that's picking out skin-specific ones and heart-specific ones. So sparsity can help create parts-based interpretable decompositions. Okay, so I said I'd say a bit more about the method. So this is the method that I developed with my graduate student Wei Wang. It's in the Journal of Machine Learning Research 2021 for fitting what we call empirical Bayes matrix factorization methods. So it is based on this basic matrix factorization model with a Gaussian errors. And the key important part is that the elements of LK and the elements of FK are assigned prior distributions, which I've called GL and GF, that are specific to each K. So this allows that some components might be more sparse than others. In particular, they're a member of some family, GL, and a family, GF, which may or may not be the same family. And then we fit this model by empirical Bayes, which means we estimate the GLK and the GFK from the data, as well as estimating the Ls and the Fs. And Ideally, we might do that by maximizing the likelihood for the GLK and the GFK, marginalizing out these guys. That's not tractable, but you can do a variational approximation. So that's what we do. So you can think of this as we're estimating the GLK, GFK. If these prior families are sparse, say they're spike and slab type prior families, then what this is doing is estimating how sparse L and F may be. If these prior families are non-negative, then you're going to result in automatically in some kind of non-negative matrix decomposition. If they're non-negative and sparse, so a mixture of a point mass at 0 and an exponential, say, would be like a, quote, spike and slab, but non-negative instead of your regular spike and slab, then you'll get some kind of sparse non-negative matrix factorization. So I'll show you some results from that. So it's a whole family of different approaches depending on which prior families you fit here. And an important point of the paper is that there's a kind of a unified fitting procedure that you can use whatever the prior families are. You essentially just have to plug in one function for each prior family you want to fit, and then you can mix and match them. So you can fit models where F is sparse but not non-negative, and L is non-negative, or vice versa, or both are non-negative, or any combination of these things. So to summarize the contributions or why I like this framework, it's flexible. You can use it with lots of different prior families. It's self-tuning. That's the empirical based part. So if you try to fit a sparsity penalty, say by lasso on these models, you have to tune the penalty that you're going to use, and that's tricky. And if you have if you want to allow a separate penalty for every factor, and one for L and a different one for F, because they may not both be equally sparse, you get 2K tuning parameters, which is a real pain to tune by, say, cross-validation. So people don't do that. They usually assume at most 1 or 2 tuning parameters for the whole thing. So I think that the empirical base has some very nice, practical features aside from anything else. For self-tuning, it's scalable for large problems, and for sparse matrices, I'll just mention that my student Jason Wilverscheid did a follow-up work on this where he really worked on making the computation scale, actually, and particularly by exploiting the fact that it could deal with sparse matrices and use matrix vector products are the primary calculation that is being done repeatedly. So if your matrix is sparse, you can exploit sparse matrix libraries. Okay. Okay, so I mentioned you can do, using this framework, a sparse version of non-negative matrix factorization. That might sound kind of, I don't know, banal, because NMF was introduced 25 years ago now, and methods for inducing sparsity seem to be all over the place. So you would think that there were lots of different methods for producing sparse versions of NMF, but there aren't that many, and it's actually a really hard problem to get stuff working, is that our experience. So for various reasons, it's a lot harder than the not NMF version of matrix factorization. So there aren't that many versions of sparse NMF. We have one. And I just wanted to show you really a quick example to show that you do get, although NMF is already sparse, on these data, at least, you get representations that are quite a bit sparser than NMF. So I should say, at a very high level, convincing someone that your clustering method is better than someone else's clustering method is really hard. And convincing someone that your matrix decomposition is more interpretable or better than someone else's matrix decomposition is also a bit hard. So, I'd like to try and do that. But more than anything, I want to convince you that you get different results if you make different assumptions. And then we can argue a bit about which result is better. And in fact, at a high level, I don't necessarily think one is right and one is wrong. We'll see later examples where they're highlighting different features of the data sometimes, right? So maybe you want to use more than one method to try to see what the differences are. Anyway, so this is single cell data from mice now. So each row now is a cell, each column is a gene. And I'm not going to go into depth on this. It's going to be a bit superficial, my description of the results, but I do want to highlight that one of the main contributions of this study was to identify a new cell type that they call pulmonary ionocytes. And we'll see those in the results in a moment. So we're analyzing count data, but we're using a Gaussian model. So we do a transformation, and we'll come back to that later as well. But that's what we did here, just for completeness. It's like a standard transformation that's used in single cell analysis often, which is a shifted log, essentially. So this is the results from eBNF. So instead of showing you that bar plot now, I'm showing you a heat map, which in some ways deals better with 30 different components, although in other ways is a little bit harder to see. So it has advantages and disadvantages. So it's a different representation, but each factor or each component here has some non-negative value for each cell. And I've scaled these so the biggest value is 1 for each, I believe. So, and 1 is black, and we have different gray levels showing partial memberships. So, and then the cells, the rows of cells, and they're arranged by labels that were provided by the original authors, which are already probably based on clustering and marker gene detection. So I think are useful for laying things out, but shouldn't really be considered maybe a gold standard. But anyway, so you can see that the first factor is kind of a baseline factor. Many of the cells have some kind of loading in that factor, and then many of the other factors are quite sparse and often pick out, say, some subset of cells in a cell type. And I'll just highlight particularly this factor 20, for example, picks out a subset of the ionocytes, and so is like picking out some of those pulmonary ionocytes, maybe corresponds to the pulmonary ionocytes that the original data, the original paper identified. This is NMF. You can see it's somewhat sparse, but not as sparse. And it doesn't have a factor that's specific to the pulmonary ionocytes. Maybe the closest one is this one's sharing between neuroendocrine and ionocytes, and this one is a more broadly shared factor. There's not one that's specifically picking out the ionocytes. So maybe the sparsity is helping pick out something more important there. So as I say, the thing I can claim, and I think you'll agree with, is these are different, and this one's sparser. And then we can argue over which one's quotes more interpretable. But that argument could take a while, so I'm going to move on to talk about a bit more about this transformation that we did. So if you remember, when I started, I said 4 things I wanted to talk about that impact interpretability: non-negativity, sparsity, and then the third one was transformations or link functions. So turns out that I've shown 2 different versions of NMF. The first one was a Poisson NMF, and the second one was this transformed NMF followed by a Gaussian or Frobenius model fit to it, right? So these are quite different, and actually it turns out that there are many data sets where you could do both these analyses and they produce different results. But there are kind of theoretical arguments for both these approaches. When I say theoretical here, what I mean is I'd argue, a priori Poisson NMF might make sense because these data are counts, and a Poisson is a model for counts, so it kind of feels like that's a better model, right? But on the other hand, when we're dealing with gene expression, usually we think of what is relevant to the changes is fold change of gene expression. It's natural to think of, say, when a module comes in and affects gene expression, it's natural to think about its effect in terms of doubling gene expression or tripling gene expression rather than adding X to the gene expression of a gene. So in many ways, this transformation can be thought of as modeling something closer to multiplicative effects of modules. And so that might be preferable as well. So we can kind of combine those ideas by, we're having a Poisson model, but having a link function instead of doing the transformation. So this link function has the property that it's suitable, unlike the log link function, it keeps non-negative, it maps non-negative things to non-negative things, right? So the shifted log, whatever value of C you use here, maps non-negative lambda to non-negative lambda, and so it's kind of more suited, well, it's suited to non-negative matrix factorization in a way that, say, the log link is not. So, okay, this is actually a family now, because it depends on what value of C you choose. Right? So C, think of this as a thing you can choose, and it changes the results. If C is very big, then this will typically be small, and you get log 1 plus something small, which is something small. So when C is big, this becomes essentially the identity link up to a constant. And so actually, when C is very big, this will give you the original Poisson NMF with additive effects that I already showed, what I called a topic model. Okay? So when C is big, this model ends up giving you essentially Poisson NMF or topic modeling. Whereas when C is smaller, it gives you something that behaves a little bit more like a log, and so you get something a bit more multiplicative. And it turns out that this has a big effect on the representations you get. So, so this is another RNA sequencing data set to illustrate the difference between the two. Again, I'm going to focus on the fact that they're different and convince you that first, right? So these are pancreatic cells, and maybe I'll start with the topic model here. These cell types are also kind of labeled by a similar clustering plus, not by us, by someone else who knows what they're doing, clustering plus marker gene type things. I tend to be kind of pretty skeptical when people give me gene cell labels, but having looked at some of these, I think these are probably mostly, especially the common cell types, probably mostly pretty reliable. And you can see in any case, the matrix factorization methods do largely kind of recapitulate this cell type structure. So as we saw before, you can see that this, with the identity link, the Poisson NMF, is kind of doing something close-ish to clustering, right? The alpha cells are all mostly dominated by membership in this red cluster, the beta cells by membership in a different cluster. There is some sharing of the red across here, which might help, although there's some over here as well, I guess. But you might, and the gray here, I guess, might be capturing some commonalities among different cell types. So you get a bit of that going on, but the overwhelming picture that you're getting is, oh, there are some different cell types here, and each component is kind of picking up a cell type. Whereas in the log link for C equals 1 for illustration, it's actually my favorite value of C. If you want to ask me what value of C shall I use, I'll tell you 1. Okay. The representation is a bit more complicated, right? And maybe highlights, certainly, okay, let's look at the delta and gamma cells. We'll see these in a bit more detail in a moment. But these cells are actually very similar to one another. They only differ substantially at a few genes. And so you can kind of see a lot of that similarity coming out in the decomposition here, right? They both have appreciable membership in the red, blue, orange, and pink, but with appreciably less in the orange for the gamma than in the delta cells, and maybe more in the pink for the gamma than the delta. And we could go and look at the genes that are involved in those different factors. I'm only showing you the loadings here, right? The one side. We can go and look at the factors to see what's going on there. And okay, we've also seen that the orange and pink here are distinguishing delta and gamma cells. That's not a coincidence. We colored them orange and pink, so that in both cases, the orange and pink are distinguishing, but the factors themselves are different, right? They're being inferred. The factors inferred by this method are different than the factors inferred by this. So let's look at the orange and pink factors for this method and the orange and pink factors for this method. So let's do the topic model first. So this is the orange factor and this is the pink factor. And what you can see is that they're So there's a point here for each gene, and essentially each, the size of the point represents how strongly is this gene influenced by this factor, if you like. We're using causal language a little bit hesitantly in this audience. So what you immediately see is that a lot of the genes are, that these factors are very correlated. And that's because of what I said, basically the orange factor's capturing the delta cells, the pink's the gamma cells. To a first approximation, the orange is just the average expression of each gene in delta, and the pink is just average expression of each gene in gamma. And it turns out that the average expression of most genes in gamma is a good prediction for what it will be in delta. They're highly correlated. So that's what's going on here. And then there are a few genes that are really different. PPY is kind of the classical canonical marker gene for gamma cells. In fact, I think they're often called PP cells or PPY cells. Yeah, this is just highlighting the genes that are really quite different in gamma versus delta, but most of the factors are correlated. Whereas the picture here between the orange and the pink is very different. They're non-negative, of course, but they're pretty uncorrelated. And you can see that the pink is really dominated by the PPY gene, and the orange is dominated by the SST and the RBP4 gene, although SST shows a bit of membership in both. And so this is kind of maybe a little bit what's going on here. You're getting maybe a more clustered representation, but it's using, because it's doing that, it has to use correlated factors to do it, because it's coloring the delta ones orange, it's coloring the gamma ones pink, but the gamma and the delta are kind of similar, so it's making the orange and the pink more similar. Whereas here, they're more independent, and so there, in order to see the similarity between the delta cells and the gamma cells, it has to give them similar colors. Do you see what I mean? So in this picture, the similarity between delta and gamma is kind of hidden because you color one orange and one pink, and you hide the fact that the orange is actually really similar to the pink, whereas here it's more exposed. And this is a slightly different to whether the parts are interpretable, but is also kind of, I think, an interesting thing to keep in mind. So another way of thinking about it is that the factors here are a bit more orthogonal than here, right? So I want to go on to orthogonality now. Now we didn't impose orthogonality here. That just kind of fell out. It just happened. It just like, in a way, just like NMF tends to lead to sparser representations, even when you don't ask for sparsity explicitly. In some ways, this log-link function seems to lead to more independent factors for reasons I can't fully kind of explain, but I think is probably systematic rather than just a coincidence. But we could ask for them to be orthogonal, right? Okay, so why might we want to do that? I should say, when I started this work, I was very against orthogonality. I was like, orthogonality? That's just a mathematical convenience in PCA. Biology is not orthogonal. So why would we want to use orthogonality? And now I've backed off a bit on that. I think there are some reasons you might want to see more orthogonality. And essentially, it's that when factors are correlated, it means you can't see how similar things are from their loadings alone, the L matrix alone. And one way to address this is to decompose the covariance matrix, XX transpose, which is LL as LL transpose, instead of decomposing the data matrix X as LF transpose. And this is kind of equivalent to doing this with F transpose F equal to the identity. All right, so if you impose orthogonality on F or orthonormality here on F, then you get a decomposition that's XX transpose is approximately LF transpose FL transpose, and that's the identity, so it's approximately LL transpose. Right? So one way to do it is to decompose X as LF transpose with orthogonality on F. Another way to accomplish a similar goal is to directly decompose the covariance matrix instead. So these are closely related. And for various reasons I won't talk about, we actually do this often. Okay, a different example, not gene expression now, for those of you who don't like gene expression. This is genotype data, still genetic. So each individual, you collect their genotype data, and you're applying matrix factorization to that genotype data. So it's individuals, these are humans individuals from the 1000 Genomes Project, individual by SNP. Each entry is a 0, 1, or 2. And you apply admixture, which is a version of NMF that kind of models the 0s, 1s, and 2s instead of just Frobenius norm. So this is NMF, let's say, applied to the genotype data. And this is empirical Bayes covariance decomposition. It's a non-negative matrix factorization applied to the covariance matrix, or actually to XX transpose. So it's a bit like imposing an orthogonality constraint on the Fs. So what you see again is in this picture here, okay, so each big bar is individuals who are sampled from the same geographic location or at least have ancestry from the same geographic location. So here are the British here, just for example, Finns, Japanese, Chinese here. These are the African Americas, Europe, India, East Asia. So they're arranged by country, not country, by broad geographic, well, sometimes country, by these labels, population identifiers, and by continent, and then they're colored again with each color representing a component. So I just want to highlight, you can see it's kind of a clustery type structure. The Finns are largely given their own component, but you can see some of their shared ancestry with the Europeans with this darker blue here. And the Japanese are largely given their own component here, colored pink, to kind of highlight the fact that you can't see from this picture that the Japanese are genetically actually very similar to the Chinese, relatively speaking, compared with the other populations. And this is a decomposition of the covariance matrix, or XX transpose, actually. And what you see here is that the Finns now, well, firstly, just like we saw in the gene expression data with the log 1 transformation, everything's a bit more complicated. Everything's broken into a bit more parts. Those parts are, you can think of them as orthogonal or independent. And so that means you can essentially read off how similar pairs of samples are based on how similar their bars are. Right? So in this representation, we can see that the Finns share a lot in common with, say, the Central Europeans here, but they also share something in common with the Indian and East Asian samples with this gray bar, which is completely invisible in this representation because it was put into the other matrix that we weren't looking at. And similarly with the Japanese, you can see that the Japanese now share a lot of components in this representation with the other East Asian groups, but have, are distinguished by a greater membership in this K6 green bar. So the point here is that if you do covariance decomposition, or equivalently, you do decomposition of the data matrix with an orthogonality constraint on one of the matrices, you end up that the matrix you get represents the similarities in your data, which may or may not be helpful, but if it's helpful, then that's a good thing to do. Okay. How am I doing for time? So this is the last example, so good. Okay. So this is a slightly more extended example. All the ones that I've done so far have kind of been a bit of like, maybe superficial, you could argue, just one slide on what, and I think actually many of these, that does highlight my attitude that a lot of these methods are most useful for visualizing things and seeing patterns in data. But this was an example where we tried to argue that we can learn something maybe potentially interesting, useful medically here. So it's an analysis of single cell data from pancreatic ductal adenocarcinoma tumors. So one of the ideas here that people who had previously analyzed these data were motivated by is the idea that maybe we could use these RNA sequencing data in single cells from these tumors to identify subtypes of pancreatic cancer, or at least maybe we can find, what's the heterogeneity look like in the expression patterns of these tumors. So previous studies that applied NMF first early on to microarray expression and then later to single cell data to find subtypes of PDAC, which they claimed fell into two categories, basal and classical subtypes. And then as more studies were done, I guess, they started to wonder whether these were really just two subtypes or were there subtypes. And, I think the story's not completely understood, but so the understanding of these subtypes is evolving. So we took data from 3 studies cited here. So there's going to be maybe some potential for batch effects, I guess. That's really why I want to tell you this. There are some of them are primary tumors, some of them are metastatic tumors. They're also slightly different assays. But anyway, so we combined all these data together. This is a t-SNE plot of these data. And the point I want to make here is that firstly, things definitely cluster in these data because these cells here all came from the same patient, patient 12 in study A. And these cells all came from 6 in A, and these all came from the same patient in C. So what the main pattern in these data is actually cells from the same patient look very similar to one another. Okay, that's not maybe a surprise, and it's probably driven by copy number variation in these tumor samples. And although t-SNE isn't ideally suited to this, you can kind of see that all the C's, or most of the C's are over here, and most of the A's are over here, and most of the B's are over here. So although it's not ideally suited to this, you get the hint that there might be some study-specific effects, maybe batch effects. So both these things might be considered technical, actually. But okay. The real challenge here is that the patient effects are masking or dominating the variation and potentially masking interesting variation that's shared between patients. And that's where, so clustering these data is just going to give you the patients back. And this is why some kind of matrix decomposition might be a more useful way to understand the structure. So the method we use, which we call generalized binary covariance decomposition, combines several ideas that I've mentioned today. We transform the counts using the shifted log transform. So although we have this Poisson model with a link function now, we didn't have that when we did this work. And in any case, it's we don't have the Bayesian framework for fitting that model yet. We just did those by maximum likelihood. So that model's a lot harder to fit still than the Gaussian model. So we still use this transformation followed by a Gaussian model a lot, and we probably will continue to. Then we decompose the covariance matrix, which is like this, making sure that we get the similarities reflected in the L's, and then we estimate F, having fixed the L's, we estimate F with a sparse, but not non-negative in this case, prior on the F. So the end result is a decomposition of this form where the F's are approximately orthogonal because we kind of assumed orthogonality of the F's under the rug when we estimated the L's by doing the covariance decomposition, but then we didn't impose orthogonality when we estimate the F's. So there's kind of a hybrid going on here, but the F's are going to be approximately orthogonal. And you can think of the L's are non-negative. So you can think of the L's as capturing how strong is component K in cell I. And you can think of F as what's the impact of component K on gene J, which can be positive or negative. So the idea here is that it's a semi-non-negative matrix factorization. The cells have memberships in biological processes. Think of that as how intense is that biological process in that cell. That can't be negative. It could be 0 if it's not present, or 1 if it's maximally present, or somewhere in between. whereas the Fs represent the impact of that biological process on each gene. So those can be positive or negative because genes can go up or down as a result of biological process. So that's the interpretation of these guys. And we call these gene expression programs, although that's a kind of that's a what's the word I'm trying to find here? It's like aspirational name for, we're hoping that we're going to find gene expression programs, but some of them might actually be technical or batch effects, and we'll go and try and see those in a minute. Okay. So this is a heat map showing the 34 different GEPs we identified. Let's look at these ones on the right-hand side first. The rows here are cells, but they're organized by patient. So this pink, for example, is all cells from one pink patient, and then there's a blue and etc. Okay, so each of these colors here corresponds to a patient, and then the patients are arranged within cohorts. So cohort A, cohort B, cohort C. So you can see this 15 here is mostly on in cohort C, and 16 is mostly on in cells from cohorts A and B. So we might think of these as capturing batch effects of some kind or study effects. And then most of these ones here, and this is how we pick these out, are patient specific. So these is mostly on in the cells from this patient, and these are mostly on in cells from this patient. So not every patient has got its own factor here, but many of them do. And then these are ones, are GPs that are shared across patients. That's how we've divided these. So we are going to kind of ignore these. We kind of think of those as identifying the strong patient-specific effects. And the idea is that once it's identified these, what's left may be interesting biology that's shared across patients. And some of these GPs, the first 8 or 9, I think, are actually, many of them are correlated with what people have previously described as classical or basal subtypes. And then the 10 to 14 are not so correlated. So that's what this plot is showing. These are different authors' definitions of subtypes of classical or basal, or even a couple of other different subtypes they've given names to in the literature. And this is just showing how correlated our GEPs are with those subtypes. You can see each of 1 to 9 are correlated to some extent with one of the previously, or one or more of the previously described subtypes, whereas 9, I think it's 9 to 14 are not. So we focused a bit more on 9 to 14, and we asked, well, which genes are involved? And when you look at the gene sets that are enriched, these are the genes that show the strongest change associated with those factors. And these are results of gene set enrichment type analyses to see which pathways these genes tend to occur in, or which gene sets these genes tend to occur in. And all of these are more or less potentially interesting. I don't have time to go through all of them, but I'd say we and our co-authors were particularly interested in this one, which shows enrichment among genes that are upregulated in response to hypoxia and more generally the integrated stress response. And I guess we're particularly interested in it because it turned out that when we tried looking at whether these gene sets were associated with survival in bulk data, so we don't have survival information on the single cell data, but we have other studies where survival is available on bulk data. So we have to take these gene sets and their scores and somehow map them to a score in bulk data, which isn't a perfect process, but we do our best, and then create a score in the bulk data and see whether it's correlated with survival. This one showed the strongest association with survival, along with another gene program that was correlated with classical stroke basal subtypes. So this is the survival analysis results, including age and sex, which are not correlated with survival in these data, but stage of tumor is correlated with survival. Well, that's pretty reassuring, I think. And then these 2 among the GPs I mentioned, only 2 were correlated in I think we did a stepwise regression here. because we don't have a SUSY version for doing survival when we did this. We do now. So anyway, this one's potentially interesting, I guess, for further follow-up. So to summarize, I think matrix factorization is a great way to explore data sets to try to understand what's going on in this data and can provide scientific insights in a wide range of problems. But how you use it matters and affects the results. And I tend to think, going back to some of those examples, one would produce a kind of a clustering representation or a more clustered representation. It's not that that representation is wrong. It's just not highlighting certain features as well as it might, right? And so, different representations may be useful depending on what you're trying to achieve, or might highlight different features of the data. And little plug, the empirical-based matrix factorization, I think, provides a nice and flexible framework for exploring those issues. So I want to thank my co-authors who did a lot of this work in alphabetical order: Peter, Kushal, Junsuk, Yusha, Wei, Eric, and Jason, as well as some others that I don't have room to mention. The work is supported by NHGRI, and here are some references if you are interested in further reading. Thanks very much.